Good morning, everyone. I am Atsuko Miyawaki-Kuwakado from Kyushu University, Japan. Today, I will present our recent study on how climate change affects dormancy release in Somei-yosh, a major cherry blossom cultivar in Japan, using the molecular phenology approach. In Japan, cherry blossom phenology holds cultural significance. People enjoy cherry blossom viewing and celebrate school graduations and entrances by cherry blossoms. Each year, many cherry blossom forecasts are made. However, last year, the forecast was challenging. In Fukuoka, the bloom time delayed about a week. On the other hand, according to Japan Meteorological Agency, cherry blossoms have been blooming earlier, 1 week earlier compared to 70 years ago. This trend can be understood by studying the phenology of temperate fruit species. Birds enter endodormancy during autumn, requiring a period of chilling to dormancy release and transition to ecodormancy. Conversely, warmer temperatures accelerate bud development. Thus, warmer winter due to global warming will prevent dormancy release, leading to potential failures in blooming. Including dormancy release in models could improve bloom predictions. However, detecting dormancy release is challenging, and most models rely on temperature and flying data. So, to enhance prediction accuracy, it is important to understand how cherry tree buds alter their internal gene expression patterns in response to seasonal changes. Such studies applying molecular phenology in the field environment have only just begun. In Lasakare, which includes cherry trees, 6 dormancy-associated matchbox genes. DAM genes have been identified as key regulators of dormancy release. As DAM expression decreases, dormancy is released, suggesting that seasonal expression patterns could help predict its timing. However, their roles under natural field environment remain unclear. In this study, we aim to develop a genetically informed prediction model for endodormancy release focused on DAM genes. Since Somei-yosh is a clonal species planted across various latitudes in Japan, it is ideal for studying various environmental responses. First, we compared molecular phenology of Somei-yosh at 3 sites across latitude: Sapporo as north, Tsukuba as central, and Fukuoka as south. But samples were collected year-round from June to next April. This is the workflow for processing gene expression data. For analysis, we focused on about 29,000 genes expressed in birds from the maternal lineage. We used hierarchical clustering to group similar expression patterns and identified 5 transcriptional modes, or subjective seasons: early summer, summer, autumn, winter, and spring. Notably, the summer mode was absent. In north, temperatures remain cool even in summer, under 30 degrees Celsius. It suggests that subjective season may correspond to temperature. To examine whether these subjective seasons correspond to temperature, we performed a number. In the winter and spring modes, the temperature was almost below 10 degrees Celsius, in contrast to other modes. However, no distinct separation was observed when examining their length. These results suggest that temperature is a major determinant of subjective. Also, hierarchical clustering revealed 6 gene sets representing the 5 subjective seasons. In the left figure, red shows high expression and blue shows low expression. The right figure shows the time course of z-score means for each site. DAM1 and DAM2 peaked in early summer and were lowest in winter, while DAM3 to 6 peaked in winter and were lowest in spring. Taking a closer look at the DAM genes, DAM1 and DAM2, colored in red and yellow, were highly expressed in summer. DAM3, colored in green, showed 2 expression peaks, while DAM4 to DAM6, colored in blue, purple, and pink, peaked in early winter and decreased towards spring. Specifically, DAM4 was suppressed earlier in north than in other sites, dropping to nearly zero by February, while in central and south, this occurred in March. As DAM expression decreases, dormancy release happens, suggesting earlier dormancy release in north. To confirm this trend, we conducted both experiments, collecting 3 branches from 4 trees in the field at the same 3 sites, north, central, and south, from October to the next March. After incubating them under constant temperature and day length for 20 days, we recorded birth break, 1 for burst and 0 for no burst. These figures show the time course of the birth break at 3 sites, reflecting dormancy release timing. Dormancy release occurred before February in north, but before March in central and south. These results suggest that the seasonal dynamics of DAM4 to DAM6 expression may provide a rough estimate of dormancy release timing. We then performed logistic that damn-for expression best reflects dormancy release. Building upon Jean's findings, we developed a method to predict the timing of dormancy release based on damn-for expression pattern. Here is an outline of the model. First, we determined the damn-for expression threshold at which dormancy release occurs. Second, we estimated the temperature threshold at which damn-for expression starts to decrease from the peak. Third, we estimated the number of chilling days required to reduce damn-for expression below the threshold. First, using logistic regression, we estimated the damn-for expression threshold for butt break probability of 0.5. Second, we estimated the temperature threshold for damp-for suppression, defined as a 7-day average temperature around peak damp-for expression, about 10 degrees Celsius. Third, using the decay equation for damp-for expression, we calculated it takes chilling 61.1 days that the temperature drops below the threshold. Finally, we forecasted the timing of dormancy release from 1952 to 2025 using daily mean temperature data and thresholds. For example, in South 2019, we counted cumulative days below 11.3 degrees Celsius from August to next July. Dormancy release was predicted on February 11th, when the count reached 62 days. Here is the estimated timing of dormancy release. The left panel shows the cumulative days, and the right panel shows the dormancy release date for each site. Past years are colored in blue, and present years are colored in red. Notably, dormancy release has delayed by 2.3 days per decade. For example, in South, dormancy release was delayed by 6.4 days over 30 years. On the other hand, the flowering date advanced by 1.9 days per decade. The diamonds represent the timing of flowering from 1953 to 2025 according to Japan Meteorological Agency. For example, in South, flowering was advanced by 7.0 days over 30 years. When we focus on the interval between dormancy release and flowering dates, it has been shortened by 4.2 days per decade. For example, in Fukuoka, the interval shortened by 13.4 days over 30 years. These results indicate that as global warming advances, the interval will shorten, making flowering harder, especially in southern part of Japan. In summary, we developed a model predicting dormancy release based on damper expression levels, which will be useful to evaluate the impact of climate change on vertebrate phenology. The model predicts a 1-week delay in dormancy release in 2024, likely due to a warm winter, which also delayed flowering. To enhance the accuracy of endodormancy release prediction, more data is needed. We collected samples at 1 to 2 week intervals throughout the dormancy season. DAM4 to DAM6 showed expression patterns consistent with the previous study. Moving forward, we are planning to incorporate these expression patterns into our endodormancy release prediction model. I would like to thank my collaborators